It's okay, quick review of the forthcoming lab on vitamin C analysis by HPLC. What we're going to be covering, you'll have covered some of this in previous modules such as biochemistry where you've done calibration lines. Okay, chromatography, a wide-ranging group of methods used for all sorts of things. The word chromatography is derived from these Greek words, chroma and graphene. It's essentially about the separation of mixtures. In this example of a technique known as column chromatography, the different components are coloured. This is nice, but often not the case. For example, vitamin C is colourless. There are ways around this. All chromatographic methods have these characteristics in common. They have a stationary phase, a mobile phase, a sample, and some way of detecting the separated components. So a little bit more detail on these. They all have some sort of solid stationary phase, which may be something like paper, or maybe a finely divided powder like in HPLC. Something moves through that. It could be a fluid or a gas. It separates out the analytes of interest, and we've arranged different methods to detect them as they come through. Okay, whip through there. Sorry, I didn't know I was doing that. Here's an example. So have a brief think about how these characteristics apply to primer chromatography. Maybe pause the video. Okay, I'm going to assume we've had a pause. Primer chromatography is the simplest system. Here samples are dotted onto the bottom of a piece of paper, the stationary phase, which is then placed in a solvent, often water. The solvent, that is to say the mobile phase, moves up the paper, separating the components as it does so. Here we can detect the separated components visually and probably measure them with a ruler. This is a typical paper chromatography results. By itself it doesn't mean very little, so we need to use standards. Again, you probably know this from certainly qualitative and quantitative analysis in biochemistry. Analysing proteins, for example. We need examples of known compounds of interest to compare with, what we call standards. This is just a description of how the process works. We start with the samples on the bottom of the piece of paper. The mobile phase moves up the paper, taking the samples with us, and we end up with a completed chromatogram. So we're going to be doing this using the HPLC machine, which does all this for us, in a sense, anyway. Since all versions of experiment differ in some ways, we characterise the separations by measuring the separated distance from a spot as a percentage of the distance to solvent front, called the RF value. In HPLC, we talk about the retention time, which more in a little while. But here's a fact that developing a method always takes much longer than running it later, and any of you do sort of this stuff for your final year project, for example, will be spending most of your time developing the method, and then when you've got that sorted out, the analyses run very quickly normally. One thing we always need to do is separate what we're interested in from the matrix which it contains. In this case, an orange. To analyse the vitamin C, we need to extract it from the orange. Then we analyse using HPLC. This is quite easy because ascorbic acid is soluble in water, that's vitamin C. We use a food blender to produce orange juice. We need to be careful not to heat the sample up too much because vitamin C is damaged by heat. We pipette a known volume of the juice, transfer it to a volumetric flask and make it up a known volume. We can't put this in the HPLC machine straight away because it contains solids, so we often use a syringe filter as is shown in the picture there. Produce a sample that contains no solids. This can then be analysed by HPLC. Okay, HPLC. High performance liquid chromatography or high pressure liquid chromatography using these types of columns we see illustrated here. It's a key method for instrumental chemical analysis and probably the most widely used chemical analysis technique. It uses a finely divided solid separating matrix containing metal tubes, that's the stationary phase, and the mobile phase is pushed through the column under high pressure and at very accurate flow rates as well. It only uses a tiny amount of sample, which can often be quite important. It probably doesn't matter so much when we're analysing routine materials in food, but things that are very expensive, this can be a very important characteristic. Says there in one of the bullet points, it's computer controlled. And it is. And we can do multi-sample analysis in principle using equipment we have. We can do 100 samples. We can set 100 samples going and come back in a couple of days and get the results. We're not going to do that, but it's certainly possible to do that. Okay, so here's a schematic diagram of a typical HPLC system. It's all sort of folded away often. It's a little bit hard to see what's happening. The mobile phase is containing one or more reservoirs. This uses to either use a single mobile phase or have more complex mixtures to be used, which we can change during the course of the experiment. In this case, for this analysis of vitamin C, we're just using a single mobile phase. It then goes to a very accurate pump, but before it reaches that, it has to be degassed. HPLC columns are very tightly packed and can be blocked by small bubbles of air. A vacuum degasser removes any atmospheric gases from the mobile phase. It'll be the noise you hear when you're using the HPLC. Historically, we would inject using a loop system with a hypodermic syringe. These days we use an autosampler, as I mentioned. You'll be sure how that works in the lab. There's characteristics of the column. Pressure's quite high. These columns, depending on what they are, some of the ones we use are around 20 centimetres in length for this experiment. And you have to have silica, that is to say finely divided sand, which has carbon chains bonded, of which more in a moment. And there's some examples. We use a C18 column for this analysis. This type of column is often used for small molecules like vitamin C or caffeine. C8 columns are used for larger molecules such as peptides, and then there's a range of other more exotic materials used. A very large number of analyses can be done. The chromatogram, and that's a nice looking one, has a retention time along the x-axis and a peak area on the y-axis, and they all look fairly similar. The retention time, which is a qualitative measure, it indicates what compounds are present but tells us nothing about their concentrations. We get that information from the peak area. Typically we run standards, in this case of different known concentrations of vitamin C which you'll develop in the lab. These are then used to develop a calibration line in a way which you will be familiar with. Finally mentioned the detector. We're using a UV detector at a particular wavelength, which we'll talk about in the lab. So I mentioned vitamin C is colourless, but it does absorb in the UV region. Okay, so calibration. A little bit more on calibration. This is the process. As I mentioned, it should be familiar to you from previous experiments in biochemistry. Just to take a little walk through it though, is a typical HPLC result for sugars in orange juice. The peaks, we don't know what they are as they stand, as they've come off the machine. In order to find out what they are, we need to run some standards. Again, we see the distinction between qualitative and quantitative data here. So what we do is run a standard and identify the retention time of each of the expected components. So in this case, sucrose comes off at just under 8 minutes. So we can say with a reasonable degree of certainty that a peak at around the same time is going to be sucrose. Not necessarily true, but we tend to optimize the experiment so that this is the case. Yep, so that's highlighted that. In food, it's unlikely you'll be able to identify all of the peaks, and we don't worry about that. In this case, in vitamin C from orange juice, you'll see a lot of peaks, which only one is of any interest. So produce a standard line. Bigger peak areas are related to sample concentration. If you remember, you see anything in square brackets, it means concentration of. Produce a standard line of, in this case, for this example, sucrose concentration against peak area. Hopefully get a nice straight line. If you get a curved line, we may have to think about the concentrations and the standards we're using. So it won't be particularly reliable. Then we put a trend line through the data, we get the equation for the line, which you then rearrange to solve for x. We'll talk about this in the lab. So here's the chromatogram of a typical standard. I don't know what it's of, it's probably of something like vitamin C, and we get a nice single peak at around 15.9 minutes. Okay, this is the chromatogram for a real food sample, and it's much more complicated, but we can easily find the peak of interest based on its RT. So again, around 15.9 minutes. The system will produce a lot of data, and we need to be aware that what we're interested in is the peak area at a particular retention time. So in this case 541, let's bring that up, oops, yeah, 541.94 milliamperes at 15.9 minutes. Right, okay, so we'll talk about all this in the lab. So if it's a little bit sort of brisk there, don't worry, we'll have plenty of time to do this, and you'll have plenty of time for questions then. Okay, I'll see you later.